Welcome to this lecture on control of gene expression at transcription and translation level. What we're going to cover in this small lecture is prokaryotic genes, eukaryotic genes and the difference between the two. First we will see prokaryotic genes. Do you know what is gene? Gene is nothing but the segment of DNA. When this segment of DNA transcribe to make mRNA, after that it is translated to make functional protein which is responsible to perform function in our body. This process is known as gene expression. As you can see in this GIF, the process is explained very well mannered. After that, we will see regulatory genes. There are two types of regulatory genes. First one cis and second one is trans. Cis genes are nothing but the segment of DNA or the part of DNA. We can see. And trans genes are not the part of the DNA. They come from outside. So, the example of the cis regulatory genes are promoter and operator. As you can see, these are two cis genes. And activator and repressor are the trans regulatory genes. When activator bind to the operator, the transcription of the genes takes place. This process is known as positive regulation. Whereas when repressor bound to the operator, no transcription of genes takes place. So this process is known as negative regulation. Next, we will see Operon model. But before moving to it, we will see what is Operon exactly. So the concept is given by Jacob and Monod, and the concept tell us about how gene regulation is done at transcription level in bacteria. And it is act as a single unit. Next we will see Lac Operon, the most famous operon till now, and the question is asked is 2 marks or 4 marks, so it is very important. One fixed question come from this operon. Now Lac stand for the lactose. Lactose act as a inducer which induces the gene expression in prokaryote. That's why it is known as inducible operon. When we look at the Lac Operon, we can find out the three structural gene, operator and promoter. In this operon, lactose is converted into its monomer, that is glucose and galactose. And lactose, as we know, it is a disaccharide, whereas glucose and galactose are the monosaccharide. Next, as we know, the prokaryotic genes. The prokaryotic genes are polycistronic or multigenic. That means many proteins or more than one protein is produced from the single mRNA. Okay. After that, we will see Lac Operon. First is the I. Gene I is nothing but the repressor protein. So, gene I transcribed to make repressor mRNA, then it is translated to make repressor protein. This repressor go and bind to the operator region. So as it is bound to the operator region, RNA polymerase do not able to bind the promoter region. That's why no transcription of the structural gene takes place. And it is done in the absence of inducer, and in this case inducer is lactose. But when the inducer is present, what happens? This inducer go and bind to the repressor protein, hence make it inactive. That's why it won't able to bind the operator region. That's why now RNA polymerase come and bind to the promoter region and transcription of structural gene takes place. So Z, Y, A, that means like Z, Y and A are the structural genes which encoded different enzymes. These are these enzymes. We will see the role in detail in the next slide. These are the structural gene. First we will see LAC-Z, that is beta galactosidase. What it does is it converts lactose to its monomeric monomer, that is glucose and galactose. When it comes to permease, that means LAC-Y is nothing but the permease. It acts as a transporter which take up lactose into the cell or glucose into the cell. Then lastly we have LAC-A, which is known as transacetylase. From the name itself you get to know it transfer acetyl group from acetyl CoA to beta galactosidase. It has no role in lactose metabolism. Please take a note of the same. Okay. So we will see beta galactosidase role in detail. The lactose catabolize, that means it is break down in its monomer, that is glucose and galactose. Whereas another work done by the beta galactosidase is isomerization of lactose to its analog, that is allolactose. Next we will see the Lac Operon when it is active. Okay. But before moving to Lac Operon, we will take the example of bacteria which is growing in the medium which contain glucose and lactose. Okay, and this is the bacteria. So first preference given to glucose. It will take up glucose from the medium. Okay, but why is it so? Because glucose is the monomer or monosaccharide and it is easily metabolized, right? It won't have to metabolize into its simpler form because glucose is itself a simpler form, right? But when the glucose is depleted, that means it is over from the medium, that times another choice come to the bacteria is lactose, right? That time what it does is it take up lactose, break it down to its simpler form, that is glucose and galactose. That time Lac Operon gets activated. Hope that's clear. Okay, so let's take a quick look at the operon. First, as we know, the medium have the lactose and glucose, so it will take glucose. The first preference to glucose. Glucose inhibit the activity of adenylate cyclase, which is an enzyme which responsible for conversion of ATP to cyclic AMP. So from here you will get to know when glucose is high, cyclic AMP is low. Okay. So if cyclic AMP is not there, it won't able to bind the inactive activator protein, and hence the activator protein kept in the inactive state. So now Lac repressor protein is active. It is made from the Lac repressor gene, and it go and bind to the operator.